Neurons transmit information via electrical impulses by firing a synapse that sends neurotransmitters throughout the body. Some common neurotransmitters include serotonin, dopamine, and acetylcholine, which are present in high levels to transmit information throughout the body. But what could be present in levels higher than these neurotransmitters? The answer to that question is glutamate, which is present in levels 10,000 times higher than that of these neurotransmitters. Glutamate is an excitatory amino acid that primarily regulates rapid depolarization in the central nervous system. First, understand that depolarization occurs when the release of glutamate activates and opens an ion channel. As these cations flow into the cell becomes more depolarized. Glutamate is also known as an excitotoxin because extreme rapid depolarization can overexcite its receptors, which can be very toxic to neurons. In this video, we will introduce glutamate's excitotoxic mechanism and explore a glutamic neurotransmitter called GLT1, which may provide a way to regulate overactivation. The mechanism behind overactivation is well understood. Generally, it can occur in two different ways. Either too much glutamate is present, or the glutamate receptors are too sensitive. Either way, these conditions will rush calcium ions into the postsynaptic cell. And this is dangerous because high calcium ion concentrations activate enzymes that damage cell structures and feed into a cascade of cell degradation and death. For example, mitochondria usually need calcium ions to produce ATP, but a calcium ion overload causes a release of caspase cofactors leading to apoptosis. The presence of glutamate generally causes no problems unless the receptors are overactivated. This can cause stroke and neuronal degeneration. For example, this is a coronal slice of a mouse brain acutely injected with glutamate and stained with DAPI. The lighter stained asymmetry shows the areas of stroke. The arrows show some, but not all stroke sites. There is currently no way to specifically target the glutamate receptors, but there may be a way to regulate the transport of glutamate to make sure that glutamate levels are not toxic. Glutamate transport can be controlled by regulating glutamate transporter 1, or GLT1. GLT1 is the major glutamate transporter expressed in astroglial cells and is responsible for 90% of the total glutamate uptake. Now we are going to explore a pathway that regulates GLT1. GLT1 regulates glutamate, which regulates overactivation. GLT1 can be downregulated by activating protein kinase C. First, 4-bol esters activate protein kinase C. Then, protein kinase C phosphorylates GLT1. Phosphorylated GLT1 is now deactivated and its expression has been decreased. There are some limitations of using GLT1. GLT1 is only a method for decreasing glutamate and does not directly treat excitotoxicity. But moreover, this mechanism does not address the other cause of overactivation, which is increased sensitivity of the glutamate receptors. Also, upregulating GLT1 to decrease glutamate could affect other biological processes. Recently, scientists have discovered another potential treatment, although most findings are currently preliminary and hypothetical. This drug is a beta-lactam antibiotic, ceftriaxone. It could be administered after excitotoxicity to minimize neuron damage. The exact mechanism is unclear, but the idea behind ceftriaxone is as follows. Excitotoxicity damages GLT1, ceftriaxone upregulates GLT1 expression, and this is hypothesized to promote neuron viability. Jagada Pillai et al. Showed that ceftriaxone was able to increase GLT1 expression in cells that underwent hypoxia-induced cell death. Future research directions for excitotoxicity therapy should investigate this mechanism to determine how it may be implemented. There are some limitations of ceftriaxone as well. Ceftriaxone does not prevent overactivation, but it does provide a way to minimize neuron damage from excitotoxicity. Unfortunately, this is only a hypothetical mechanism, and many details are left unknown. It is unclear what risk factors make someone more prone to overactivation, and we also do not know biochemically how neurons prevent excitotoxicity. Nonetheless, understanding the mechanisms of glutamate regulation can lead to more effective treatment options and eventually a better understanding of the neurodegenerative diseases whose general onset still remain a mystery. There is still much left unknown, but that is the allure of science.